I think we might kick off the seminar. It's my pleasure to welcome you to the Coffee Science Seminar today. My name is Graham Hammer. I'll be hosting the session today. I'm a professor in crop science in the Centre for Crop Science in Coffee, and I work in the space of crop physiology and modelling, and as part of the ARC Centre of Excellence for Plant Success in Nature and Agriculture, which is led by Professor Christine Beveridge in Biological Sciences at UQ. It's my pleasure to welcome you today and initially acknowledge the traditional owners of the country. The University of Queensland acknowledges the traditional owners and their custodianships of the lands on which we meet. I'm currently sitting in Indooroopilly, which is a delightful Aboriginal word meaning gully of leeches, but it's quite a pleasant place to be. We pay our respects to the ancestors and their descendants who continue cultural and spiritual connections to country. The traditional owners of where I am are the Jagera and Turrbal peoples. We recognise their valuable contributions to Australian and global society. If I can just give a little bit of housekeeping. The seminar is scheduled to go from 12 to 1. We will have a question and answer session after the end of the formal presentation. And if you want to ask questions, please put them in the Q&A tab, not the chat tab. I will be monitoring the Q&A tab, and we'll have a Q&A session at the end of the seminar. So it's my pleasure to introduce Andrew Borrell for this seminar. And Andy is a crop physiologist and professor in crop science with Coffee. He's based with the UQ group at the Queensland Department of Agriculture and Fisheries Hermitage Research Facility, where he's been based for quite some time. He's worked extensively on drought adaptation and water relations in crops, and is particularly renowned for his research on the physiology and genetics of the stay green trait in sorghum. So it's my pleasure to hand it over to you, Andy.
Great. Thanks, Graham. I'm going to give an overview today of our ARC Discovery Project, and it's titled Serial Blueprints for a Water Limited World. And I'd just like to begin by introducing some of the team. So working with me on this project has been Ian Godwin and Lee Hickey as CIs from UQ, Rod Snowden from Justus Liebig University in Germany, Ben Travascus has been a PI from CSIRO, and Karen Maisel, who's been a postdoc on this project, has done a fair bit of the work based at UQ. We've had a couple of PhDs, Albert Wong and Yasmin Lam, a couple of MSc students, Jasmine Dio and Zach Eldis, Nicholas Lester did his honours work, and Eric Van Oostrom has helped to supervise both of the PhD students. So it's a great team. It's all about growing crops in a highly variable environment. And these are some slides that I took in the Darling Downs, where I live in Australia, between October and December in 2019. And you can see on the left that this is the Dam and Hermitage Research Facility, which is almost completely empty in that severe drought. You look across the landscape and you can see smoke from fires that are about 30 kilometres away. On the right-hand side, that's the Condamine River, which runs through the middle of Warwick, and at that time you could walk across it down from where I live. Wind forward less than 3 years, and these are some photos I took in April of this year. You can see the Condamine River in full flood, and on the right-hand side, that photograph there is the same stretch that I could have walked across 3 years ago, and they're at some couple of 100 meters wide. So the point is that it's an extremely variable environment in which we're living, and farmers have to contend with this and master growing crops in a highly variable environment. A bit about the conceptual framework of the Discovery Project. We're assessing the role of 2 gene families, PIN and VRN, in 2 model cereal crops, that's sorghum and barley, to better understand how these genes might regulate above and below ground plant architecture, and thereby potentially enhancing resistance to abiotic stresses such as drought and nutrients. Plant development is characterized by its flexibility and adaptability, which allow the optimal adjustment of plant shape according to the environment. And this is just one example here on the right-hand side of some T1 transgenics and the up and down regulation of SBPIN2 gene in sorghum and how that affects canopy architecture. The objectives of the ARC Discovery Project are firstly to explore the mechanisms by which PIN and VRN genes modify canopy development and root architecture. Secondly, to elucidate how above and below ground systems interact and the extent to which shoot and root systems can be modified to optimize crop adaptation to water-limited environments. And finally, to determine how the expression of PIN and VRN genes interact with particular resource-limited environments, for example, limited water and nutrients, and thereby evaluate G/E interactions. Why the PIN genes? Well, there's a few reasons why we wanted to look at the PIN family of genes. Members of the PIN family of auxin efflux carriers often appear within stay green QTL. Now, stay green is a drought adaptation trait, and I'll explain what that is in a minute. 9 of 11 genes from the PIN family in sorghum co-locate with stay green QTL. So you can see in this schematic 10 sorghum chromosomes, and you can see that the purple boxes are stay green QTL that are co-located with particular SB-PIN genes. We have been interested in a couple of these regions in particular, and I've highlighted the stay green 1 QTL, which contains 2 of these PIN genes, SB-PIN3 and SB-PIN4, and the stay green QTL, which is 2 QTL, which contains SB-PIN2. And both of those QTLs we've done detailed physiological dissection of. There's also stay green 3B that contains SB-PIN1, and that's a region that the sorghum breeding program, the public sector breeding program in Australia, have been selecting for in particular. A little bit about the stay green drought adaptation trait for those of you who are not familiar with it. If you take a senescent line and you introgress a single stay green QTL into that line, the phenotype will exhibit reduced tillering, smaller upper leaves, and this results in a smaller plant size at flowering. This leads to increased water availability at flowering, increased water use during grain fill, and also the root angle is narrow, which leads to increased water accessibility at flowering, and this can lead to increased grain yield. Now, some of these components relate to water demand. So those factors that contribute to a smaller canopy will result in less water demand. And then the other factors here that are contributing to increased water supply during grain filling period and the narrow root angle going deeper to access more water, they result in increased water supply. And together these things contribute to increased grain yield. But PIN genes are also very important in plant growth and development. And in fact, they have driven evolutionary innovations. So PIN genes underwent 3 critical evolutionary innovations and thus acquired a triple function to regulate the development of 3 essential components of the flowering plant Arabidopsis. They impacted shoot root development, inflorescence development, and floral organ development. And you can see here that particular PIN genes or combinations of PIN genes are responsible for these particular innovations. So the PIN-dependent auxin distribution system displays an extensive plasticity at the subcellular level. It represents an entry point for both environmental, and I mean things such as gravity, as well as developmental signals, which can modulate the polarity of PIN localization and hence the direction of auxin flow. And in our discovery project, if you have a look at the diagram here, you can see that the SB PIN1, the non-canonical, this is the red part here, and these non-canonical, the SB PIN1 spans the endoplasmic reticulum, and it's important for the movement in and out of auxin from the endoplasmic reticulum, and therefore has a role in auxin homeostasis within the cell. The SB-PIN2 gene is canonical, it's monocot specific, and it spans the plasma lemma membrane, and it's allowing auxin to move from the cytoplasm into the apoplast. And SB-PIN4 is semi-canonical, it's monocot specific, and it has similar functions, both the functions of SB-PIN1 and PIN2. So in the wall of the endoplasmic reticulum and crossing the plasma membrane. So PIN genes regulate canopy development in sorghum. And again, we did this work firstly back in 2017, and this is one of the reasons why we wanted to pursue it, the PIN genes in the discovery project. So you can see here that the control, Texas 430, is a medium tillering type. Downregulation of SB-PIN2 results in a lower column number per plant, and upregulation of SB-PIN2 results in a high tillering phenotype. They also impact root architecture, and you can see here that Texas 430 has got a medium root number, and downregulation of SB-PIN2 results in a low number of nodal roots, whereas upregulation of SB-PIN2 is resulting in a large root number. So why the vernalization genes? Well, many varieties of wheat and barley require prolonged exposure to cold during winter in order to flower, and that's called a vernalization response. And in these cereals, vernalization-induced flowering is controlled by the vernalization 1 or the VERN1 gene. So VERN1 is a promoter of flowering that's activated by low temperatures. Now, there was a paper recently published by Vosfels et al. In 2018, and they found that in addition to regulating flowering behavior, that these VERN genes also modulate root architecture in wheat and barley. So we thought, well, that's very interesting, and perhaps in addition to looking at what the PIN genes do, we would like to know what the VERN genes might do in terms of root and perhaps shoot architecture in barley and sorghum. So that was the thinking behind why we would look at these two families of genes. 4 hypotheses for the project. Firstly, PIN genes are major determinants of plant architecture. Secondly, that VERN1 is a major determinant of plant architecture. Thirdly, root and shoot system architecture can be modified using PIN and VERN1 combinations. And then finally, deployment of PIN and VERN genes will lead to designer idiotypes for target environments. Now, a lot of this project was about developing genetic resources. If you are an Arabidopsis scientist, then this may seem like quite a straightforward thing to do. You just go to the mutant library and you choose your gene of preference. However, if you're working with sorghum and barley, this is much more difficult to do. So the development of genetic resources took about half of this project. So the first 18 months or so was dedicated to this. And the task was largely undertaken by Karen Maisel, the postdoc, in conjunction with the PhD students. And in sorghum, the PIN overexpression lines were initially developed by Lou, who works for Ian Godwin's group, back in 2017, and Lou and I phenotyped the T1 generation. And then Albert Wong, in his PhD, progressed the development of that work into the T2 generation and the phenotyping of the T2 generation. The PIN knockout lines, Albert developed some of those within his PhD. Jasmine within her MSc with the help of Karen, and the VRN overexpression and knockout lines were developed by Karen and Yasmin as part of Yasmin's PhD. For the barley lines, PIN overexpression lines were developed by Karen, the PIN knockout lines by Karen and part of Zach's honours project, and the VRN overexpression knockout lines developed by Karen and Yasmin as part of Yasmin's PhD. So that was a very important and quite extensive part of the project. I've got a matrix here to explain how I'm going to go through the data. I'm going to look at the data by crop and gene. So there's just a matrix of crop, that's sorghum and barley, and then the gene families of PIN and VRN. And then we'll have a look at the PIN genes in barley. We'll then progress to the VRN genes in barley, and then the VRN genes in sorghum. But I'll spend most of the time looking at the PIN genes in sorghum because that's the majority of the data that we have at this time. And I want to begin by looking at the overexpression of the PIN genes in sorghum. And this is a part of Albert Wong's PhD program. So SB-PIN1, SB-PIN2, and SB-PIN4 tissue-specific expressions in sorghum were looked at by Albert. And you can see the tissues here that he sampled. The fifth fully expanded leaf that you can see circled in red. There's a 1 centimetre section of the root apical meristem, and then there's a nodal root, including the nodal root tip. This is some of the expression data that was collected. It's relative to Texas 430, so this is the baseline here, and you can see the phenotype of Texas 430 late in grain fill. Upregulation of SB-PIN2 in the meristem at 28 days after emergence was associated with a high tillering phenotype that you can see here. Downregulation of SB-PIN2 in the meristem at 28 days after emergence was associated with a low tillering, in some cases, unicolm phenotype. And if you plot the expression of SB-PIN2 relative to Texas 430 in the meristem at 28 days after emergence against column number per plant, you can see that there's a close relationship suggesting that the levels of expression there are driving this branching phenotype. So the results from our expression studies show that expression of PIN genes from a stay green source in a non-stay green genotype induce phenotypes that are differentially expressed in stay green versus non-stay green genotypes. And the result can be interpreted as showing firstly that modulation of PIN expression can mimic stay green phenotypes, and this is of practical utility, and possibly that differences in PIN gene expression or function is the basis of stay green versus non-stay green phenotypes in the corresponding genotypes. Now, we've just published this recently in JXB. SB-PINs impact canopy development, and you can see here an array of plants that have got their T2 generation overexpression transgenics that were developed by Albert. Texas 430 on the left has got that medium type of phenotype. And if you look at PIN 212 again as an example, you can see that PIN 212 is a high branching phenotype and PIN 214 is a low branching phenotype. And this is very similar to the T1 results that I showed earlier. So SB pins impact canopy development. And you can see here pin 430 with the column number here of between 4 and 5. And you can see with pin 212 that it's much higher branching, significantly higher branching, and pin 214 and pin 43 lower branching. Think about a canopy of a plant, crop like sorghum, and the canopy is a combination of the number of branches, the number of leaves per branch, and the size of the individual leaves. And you can see here that not only are the pin genes impacting column number, they're also impacting the leaf size. And there's a leaf length distribution here. And you can see that the blue line here is Texas 430, the control. But in this case, you can see pin 43 is producing longer leaves relative to the control. When you look at leaf width, you can see again the control in blue, but you can see that pin 214 and in particular pin 212 have narrower leaves. So you can see that various pin genes have an impact on making leaves larger or smaller. So together with the differences in tillering, they can have quite an impact on the size of canopies. This is a slide on the demand index and tillering. And on the x-axis, the demand index, it's really an index for a similar demand, and it's the product of leaf appearance rate and of the average area of leaves on the main stem. And for genotypes that lie on this line here, which is aggression between the demand index and between tillering, then the differences in tillering here are largely explained by differences in the demand index and a similar demand. However, sometimes genotypes lie off this regression line, either above or below, indicating that something other than demand for assimilates is driving this relationship. And we're suggesting here that it could be a hormonal impact. So SB pins impact root architecture. And you can see here some very different root architectures. So Texas 430 here has a medium biomass and a median number of nodal roots. Pin 212 has got a much larger root system and a high number of nodal roots. Pin 214 has got a smaller root system and a much smaller number of nodal roots. And SB pin 4 impacts root gravitropism. Now, you wondered about this from reading about the impact of pin genes on root angle in other crops, such as rice, and in the model plant Arabidopsis. We thought that it's likely that one of the SB pins would have an effect on gravitropism. This is a system here developed by Graham and Vijaya, in which there's 50 glass plates sitting inside one of these tubs, and you can have 10 tubs, so you can screen up to 500 plants. Albert was just looking at one tub here of 50 plates and looking at some of the pin genes. The plates can be removed. The plates are then put into a box like this, and they are imaged on both sides, and you come up with images like this. And you can see here that pin 48 has got a narrower angle than the control Texas 430. And when you look at the data, Texas 430 has got an angle of something like 35 degrees, and pin 48 something like 25 degrees. So significantly narrower angle in pin 48. There was some complementary work that was done on a Gates Foundation project that we have had in Africa, working with Jimma University. And this is Temesgen, who is one of my PhD students. And the Ethiopian Institute of Biodiversity have a wonderful collection of sorghum, about 10,000 lines. And we subsampled that back to about 2,000 lines that were representative of sorghum plants in Ethiopia. Then Temesgen, he phenotyped these. Now you can see that there's a phenotyping system similar to that which we used in Australia. So this system was actually, we built another one, transported it over to Ethiopia, and Temesgen did the work over there. And he measured the root angle on nearly 2,000 lines. He then took about half of those, about 900 lines, and he grew them out in 2 drought-limited sites in Ethiopia. And he found that narrow root angle is moderately correlated with grain yield at 2 field sites in Ethiopia. So this was a really nice link between a trait such as root angle and showing how it links through to increased grain yield under drought. SB pins impact root architecture, and this is some data from the T1 study. And you can see here on the x-axis, it's green leaf area per plant. Now, the largest contributor to this is differences in tillering, and on the y-axis, you've got the number of nodal roots per plant, which is above ground branching. So these responses from above and below ground are highly correlated and probably indicate an allometric response between above and below ground factors. Now this graph here is on the x-axis shows main shoot biomass, and the y-axis shows the number of nodal roots per reproductive column. And you might expect that as the main shoot biomass increases, so would the number of nodal roots per reproductive column, and a positive relationship, which is what occurred. But in some cases, you've got genotypes that are lying above this relationship as well, suggesting that it's something other that might be driving this process. And again, we're suggesting hormones to be involved. SB pins impact the shoot, the root to shoot interaction, and this is some data from the T2 generation collected by Elbert. And you can see here root biomass on the x-axis and shoot biomass on the y-axis. They're positively correlated, but you can see that for a given root biomass, in this case 60 grams, that in this case pin 1.6 has a shoot biomass that is twice that of pin 4.3. So for a given root biomass, there can be significant difference in shoot biomass. And these are the sorts of differences between above and below ground that we're particularly interested in if you wanted to develop idiotypes for particular target environments. This is a graph here for the T2 data, similar to the one I showed for T1, of main shoot biomass on the X and number of nodal roots per reproductive column on the Y. And again, pin 4.3 sitting up above that line. SB pins also impact inflorescence development. And you can see here there's quite an array of phenotypes for the inflorescence. Texas 430 is our standard type that we're comparing back to. Again, if we look at pin 212 and pin 214, pin 212 exhibits it's a very open panicle. And if you know anything about sorghum, it does look a bit like the Guinea types that have that very open panicle. And pin 214 is a closed panicle, higher branching density. And when you look at the data here and look at panicle branching density, you can see, in fact, that pin 214 has a significantly higher panicle branching density than does Texas 430. Albert also did some transcriptomics on the sorghum part of the work. He collected data for all of the pin genes, so from SB pin 1 through to SB pin 11 for the panicle and for the root, and these were sampled at 40 days after emergence. You can see here that downregulation of SB pin 4 in Texas 430 was associated with this type of panicle. Upregulation of SB pin 4 in pin 4.3, and this is the type of panicle that you get here. And then in the roots, this is downregulation of SB pin 4 in Texas 430, and this is a type of root system. and then upregulation of SB pin 4 in the roots in pin 4.3. And again, you can see quite a different root system. So the transcriptomics data did provide us with some insights about what particular genes and gene networks were doing. A little bit about the knockout of pin genes in sorghum. And here's Karen Maisel, who did a lot of this work. And if you think about editing of canopy-sized genes that are affecting water demand, we were expecting that we might get some differences in phenotype. So you think of a canopy, and we know from the overexpression work that we did that we get differences in the column number per plant, differences in leaves per column, and differences in the area per leaf. So we thought, yep, maybe we'll get these differences with the knockouts as well. Also, for editing of root architecture that are affecting water supply, we knew from the overexpression work that we were getting the pin genes were affecting angles of roots, elongation of roots, and branching of roots. So we thought knocking out these genes would likely give differences in phenotypes relating to these things. There was a lot of redundancy in pin gene function. So you can see here that this is a wild type, and you've got a single pin 1 and a single pin 2. So we expected phenotypic changes in pin knockouts due to, firstly, the importance of oxygen in plant growth and development, and secondly, there were many phenotypes found in the OE lines. However, limited phenotypes found in single pin knockouts. But Karen created some multi-gene CRISPR knockouts in a single generation. And you can see the wild type on the left and the single pin 1 mutant, the single pin 2 mutant, not much difference in phenotype. But you can see on the far right-hand side that the pin 1, 2 mutant does give a different phenotype. The pin 1, 2 double mutant affected leaf angle. And you can see here on the wild type that the leaves are flatter. On the pin 1, 2 double mutant, that the leaves are more erect. And you can see here that the leaf angle in the double mutant was greater than was the case for the wild type. And the double mutant also affected panicle and root architecture with a longer panel, a longer sorghum panicle for pin 1, 2, but also significant differences in root biomass length and number. It's helpful to model some of the effects of these pin genes, and Graham Hammer and Greg McLean helped to do some work on modeling the effects of SB pin 2 in both Australia and in India. I'm just going to present some data here, the modeling work, looking at a variety commonly grown in India called M35-1. And you can see here, this is a plot of days after sowing and leaf area index. The M35-1 line without the SB pin 2 effect produced a larger canopy, and with the SB pin 2 gene produced a smaller canopy that had delayed leaf senescence and a stay green phenotype here. And the next graph below shows days after sowing and extractable soil water. This is the grain filling period through here, and you can see that the line with SB pin 2 has more plant available water through that grain filling period. Now, the total biomass production was similar in both of these lines, but the harvest index was higher with SB pin 2. So you can see here the dashed line has a higher grain yield than the line without the SB pin 2. And when you do it's a type of benefit cost analysis, and you've got grain yield on the x-axis here, yield difference on the y, and you can see here that in most cases for grain yields of around 3 tons per hectare and below, that you're getting a significant advantage from having this gene in the germplasm, and then there's a small cost as you get to the higher yield end. What about PIN genes in barley? And I'll say a little bit about some riser box studies that were undertaken by Zach Aldous and by Karen Maisel. There's a lot of redundancy in the PIN genes, in the PIN gene function. You can see you've got a wild type here. You've got PIN 1, 2, 4, single mutants. They don't look very different. Maybe PIN 11, a slightly smaller canopy, but no significant differences in these above ground phenotypes. However, when you look below ground, this is the golden promise control here, and this is PIN 11 knockout edited line below. It's a -1 base pair edit. And you can see in the top one with golden promise and control that most of the roots are in the top half. And you can see with the PIN 11 knockout that most of the roots are in the top third. And in fact, there are very few roots that are in the bottom third S3. So it suggested that there's something happening with the roots and maybe there's a gravitropic response. And so this is work by Zach Aldous and Karen Maisel, showing that in fact there was a gravitropic response. This is PIN 11, it's before rotation. You can rotate at 90 degrees and then wait 24 hours, but you can see here that there's no change in the root angle. They just stay the same. With golden promise, the control, rotate at 90 degrees, and you see that the roots are bending downwards. And when you graph this out, this is the root angle on this axis here, time since rotation across here. And you can see that with golden promise, there's a change in the root angle in response to gravity and no change with the PIN 11. So a gravitropic response. The VRN genes in barley is Lee and Yasmin. And from some of the previous data, we wondered whether we get a response on root angle from VRN 1. And the experiments that we've done here, there was no difference in the seminal root angle from these various single knockouts of the VRN 1 gene. There was an impact, however, of VRN 1 on leaf length in barley. And you can see here that on some of these, you've got VRN knockouts, single VRN knockouts here, and the leaf 1 there is longer, and the same with the length of leaf 2. What about VRN genes in sorghum? So this was a bit of a question mark because sorghum does not have a vernalization response in the same way that you see in winter cereals like wheat and barley. However, this graph here shows that VRN genes were having an impact on panicle length. And just to explain this, the SB VRN 1 knockout, it's a CRISPR-Cas9 gene knockout of the sequential homologue of HV VRN 1, which is the VRN 1 gene in barley. So if you knock out the homologue of that in sorghum, then this is the line, and it produced a significantly longer panicle, as you can see over here. Now, this was measured in 2 experiments, no significance in grain yield in the first study. That study was impacted a bit had some insect damage, so the study was repeated. And in the second study, the longer panicle also exhibited a higher grain number and yield in that study. And the flowering time was delayed by about a week in the knockout lines. So to summarize, plants are sessile. They can't walk, so they've got to adapt. Genetic variation in plant architecture is required for adaptation. PIN and VRN genes have been shown to modify plant architecture. We selected model C4 at sorghum and C3 barley crops to investigate these genes. We developed overexpression lines via transformation to assess upregulation of the genes. And we knocked out these genes via CRISPR to assess loss of function. Genetic resources are currently being phenotyped for adaptation mechanisms. And PIN genes modify canopy, roots, and the panicle in sorghum. PIN genes modify root architecture in barley. VRN genes modify panicle length and grain yield in sorghum. And the VRN genes modify leaf length in barley. We had quite a few groups that invested in the work on the PIN gene transgenic studies and also the stay green research, including the root studies. So we appreciate that. Thank you.
Thanks, Andy. That's a really great presentation. Look, we have one question in the Q&A, but while there's a few more coming in, I just had a couple myself to kick it off. How do you move forward with this once you've got all these leads on various PIN genes or forms of PIN genes, and you want to, you know, you're sort of saying you might design particular trait combinations for particular environments. How do you get from this sort of a lot of work on different responses and different organs to actually heading towards something that you say, well, here's what we want?
Yeah. Well, I think we started off with an overall view or a hypothesis of what we wanted to do to look at the plasticity in above ground and below ground components. So we were particularly interested to see the extent to which we could tweak canopy development by changing the branching or the number of leaves per branch or size of leaves, and what we could do for the demand. Now, we had an idea that PIN genes would be able to do that from the earlier work. And if you also bring in the roots and then think about water supply, then the idea was, how can we, if we had a particular target environment and we wanted to develop some genotype by management combinations for that particular environment, what could the genetics look like? So I think it's getting an idea of firstly, a family of genes that underpins a stay green trait, but then bringing in another family of genes that could perturb the canopy and the roots even further. I guess to begin with, we've just looked at single genes and their impact, depending on, because of redundancy, you get an impact to some extent, but when you get double mutants or triple mutants, you can get a stronger response. What we have not done yet is combine some of the PIN and VRN gene responses to see what might happen. So I think the ultimate aim is to understand how both families of genes are operating, to look then at if we combine, if there are particular phenotypes that we're interested in, and one may come from a PIN gene, one may come from a VRN gene, are we able to bring them together? What would that phenotype look like? And initially, this will be done in transgenics, but if it's done by gene editing, hopefully we'll be able to grow those out in the field and get a wide range of phenotypes, both above and below ground, that might begin to develop into these idiotypes.
Okay, thanks. I've just got one follow-up, so excuse me if I just bombard you with a few questions first. I mean, there's a lot of pleiotropy in what you're showing there in terms of various traits being affected by different PIN genes in different ways. And, you know, we know from like the work that Christine Beveridge's work and that the group on, you know, branching in P and the sort of complexity of that network in terms of interactions of sugars with hormones, with auxins and strigolactones, and the complexity of those interactions to try to get some indication of how all that connects to link to branching. Presumably what you're sort of seeing here is some manifestation of the interaction of all these bits in a gene network. So is there any thought to thinking about trying to map out how the gene network might be working?
Yeah, the pleiotropy is interesting and might be expected because we're looking at an auxin efflux carrier, and auxin impacts the plant, everything from embryogenesis through organogenesis. It's affecting branching, the main branches, branching patterns in panicles, size of leaves, and maybe a direct and indirect effect on the roots from allometry, hormonal. So there is a lot happening, and yes, significant pleiotropic response. With the networks, we've done with the transcriptomics work in part of Albert's study on transcriptomics. We did start and we're just beginning to unwrap this at the moment, but there are some networks around the PIN genes that we can begin to explore from the transcriptomics work. But you really need to have a very specific question with transcriptomics or you can get lost, but it is giving us some understanding of the network. And then we are planning to test some simple models, maybe with Owen and Mark, just to explore this a little bit more about how it might fit into the bigger picture.
Thanks, Andy. I'll stop hogging the questions. And we have a couple in the Q&A box, and the first one is from John Dixon. I'll just read it out. Fascinating role of the PIN family genes in sorghum canopy and root development angle. Is there any info on whether PIN genes are associated with sorghum rhizome development? My interest is related to the potential of perennial sorghum.
Look,
a little bit left field.
Yeah, John, I don't know directly about the impact of PIN genes on rhizome developments and how that might affect perenniality. I know it's always interested us in the comparison between a crop like sorghum, which has a degree of perenniality, compared to wheat or barley, which have got a much stronger annual response. So when we've looked at traits like stay green, we know that it's when you're looking at age-related senescence, you've got a stronger response in wheat and barley compared to what you might have in sorghum. But as far as as you saw in the talk, there is an impact on root angle, and in Ethiopia, we've been able to show at 2 field sites that narrow root angle has contributed to higher grain yield. But as far as the impact of PIN genes on rhizome and perenniality, I don't know.
Okay. One more question from Shiva. Says, I was not clear about the difference between SB PIN 2-12 and SB PIN 2-14. We can see difference in phenotype, but what are the genetic differences? Okay,
so with their overexpression lines, the SB PIN, the PIN 212 is an upregulation of that gene, resulting in a high tillering phenotype, and in SB PIN 214, it's a downregulation of that gene that's resulting in very low tillering and in some cases a unicolm phenotype.
Okay, thanks. I think we're about done on questions, Andy. So unless there's some more to come in late, but there's no more there at the moment. So perhaps we could just wrap up if you can move on there. So thanks to everybody for joining today. You'll see the next seminar is from Ali Raza and from the Centre for Animal Science on cattle with ectoparasite resistance. And if you want to check out any more information, details about the coffee seminar, I think there's the website that you can look at that will also lead you to recordings of previous seminars and ability to get onto a mailing list if you're not already. So thanks very much for your attendance today.
Thank you.
Thanks, Andy.